So, of course, there's a lot of discussion about big data, the term big data. And one thing I would want to argue is that it's not so much about the big that is important, but it's about the complex as well. So that, in fact, complexity is as big a bottleneck in studying data as its size. So what that argues for is that one needs an organizing principle for large and complex data. And so I would argue that one version of that is that data has shape. One can organize data in terms of its shape, perhaps higher dimensional shape, and that the shape matters. Shape is one of the first things you want to know about the data, and sometimes it's enough to get you all that you need. I claim, though, that everybody in the room already knows this fact because everyone's done a regression line here. Regression line is what? It is an attempt, it is a method for replacing a data set or approximating a data set by a shape, namely a very simple shape, the line here. And when that works, that's an excellent thing to do. It provides capabilities for prediction, for overall understanding of the data, and compression. But of course, not all data fits along straight lines. The next kind of data that might come up would be a clustered decomposition like this. Data which doesn't fit along any line, but instead looks like this, where you've got three groups, perhaps each of them somehow conceptually coherent, a particular form of a disease or, or particular kinds of behaviors. In this case, the replacement, the shape that one replaces this with is, three distinct points. So it's no longer a continuous object. It's just 3 points. You might think of it as the centers of these clusters. Now, notice that the mathematics that goes with the clusters is completely different from that for linear regression, right? The clusters, it's a whole cluster analysis is a highly developed kind of chaotic field, actually, but is very rich and different, distinct from linear regression. Now, you might hope that if one hopes that all data fits along in one of these two paradigms, but then you would immediately arrive at a picture like this one, which also happens quite a bit. The data, in this case, this often comes from situations where you have periodic or recurrent behavior in the data. And the loopy aspect of this, which is the shape here, captures that or reflects that. Now, so we could say, well, let's develop another method for capturing loops. And one can do that. One might base it on Fourier analysis. One might base it on other things if the period perhaps is not constant or a very well-defined thing. But that would be yet another piece of mathematics to capture this one type of shape. And then, of course, we might then arrive at a data set that looks like this. And this also happens a lot. This is something that might happen, for example, when there's a process going on. So there are some standard behaviors which sit at the core, at the center of this. And then the three flares going out. The tips might be the ways that things go off the rails or the extreme behaviors. So now you wouldn't say, well, all right, shall we develop a method for capturing Y-junctions as well? Okay, well, we could do that, but a better idea might be to say, we're seeing all these different shapes. We actually expect to see some even more complex shapes in data going forward. And so maybe we should develop a method which can capture all shapes. Okay? And so that's the argument here. And so the question is really how to model data. There's a lot of machine learning methods around modeling data with linear methods, matrices, linear algebraic problems, sometimes quadratics and variants on that. And those, work very well in many situations. They're great. But something to capture all kinds of shapes and to sort of accurately reflect them requires a different method, which I would call network modeling, which is what we're going to talk about in the beginning today. Okay, so how is the shape of the data defined? Normally it's defined in terms of a metric on the data. We actually, I often think of the data as just finite metric spaces is the right area to work in dealing with data. It can be Euclidean distance, but there are lots of variants on that. Correlation distance, angle distance, Hamming distance when you have sequences, many other things that come up in more complex situations. The main thing about it is that the distance function should encode, in some sense, similarity. That is to say, points which are nearby should be similar in some intuitive sense. Points that are far apart should be dissimilar. And then that's kind of what one expects with these metrics. Okay. Now, so what is topology then? Topology is the formalism for doing two things. We'd argue it's for measuring and it's for representing or modeling shape. It's been part of pure math since the 1700s, but in the last 10 years, the methods from topology have been ported into this world of finite metric spaces or point clouds, and that's what we'll be talking about today. The original work on this was a recreational math problem solved by the Swiss mathematician Leonhard Euler about the bridges of Königsberg, where he asked, or the question was asked, can you cross all the bridges exactly once and get back to where you started from? And he found that the answer was no, but he did so in a very clever way. He recognized that it doesn't matter how deep the water is, how long the bridges are, how big the islands are, anything of that. It's just a simple network model for it, with 4 nodes, A, B, C, and D, and then the bridges each representing edges. With that simplified version, he was able to reason about that question very easily and solve the problem. So I would argue that topology has 3 key ideas: that it represents shape in a coordinate-free fashion, it's got invariance under deformation, and finally, and the first thing we'll talk about is compressed representations of shapes. So coordinate freeness is illustrated by this picture. A circle can obviously sit in the plane, but it can also be rotated and moved into other into space, for example. We don't care how it sits in space. We're interested in the circle itself. Invariance to deformation is a reflection of a very powerful property of the human visual system, a very fundamental property of the human visual system, which is that we can recognize letters no matter what font they're in, or whether we're seeing it at an angle or seeing it on the surface of a soccer ball. We can always recognize the A as the thing with a loop and 2 legs, and the B as the thing with 2 loops. So doing that kind of robust qualitative analysis of shapes is a big chunk of what topology is about. And we're going to try to introduce some of that into data as well. Here's a more data-centered version of that log-log plot of a circle in a plane, which is also a deformed version of the circle. And finally, the compressed representations. The circle on the left there is actually infinite amounts of information. It's infinitely many points, infinitely many pairwise distances. But if you're willing to sacrifice a little bit of detail, and often you are willing to sacrifice a little bit of detail, then you can represent it by the shape on the right, which has got the loopy aspects to it, but which has lost curvature information. And the thing on the right, of course, is a combinatorial object and can be represented very simply with 6 edges and 6 nodes, something that can be put in a single byte. Okay, so again, topology has 2 tasks, I would say: representing or modeling shape, and then measuring shape. We'll try to talk about both of them today. So here's the schematic idea for how you represent or model shape or a circle. So or one version of it. So we have the circle, but we also have some data about it. Namely, we have a covering by 3 sets: a red set, a blue set, and a yellow set. The blue set breaks up into 2 connected components, and the red and the yellow are single connected components. So a strategy for building a complex or one of these compressed representations here, it goes as follows. Take the connected components of each of these each of these sets and build a node in a network for them. Okay, so the blue one there has 2 because it's got 2 connected components. And now, because they overlap, notice the sets overlap, the red overlaps with the blue and the purple, and so on, we'll draw an edge between any pair of nodes whose corresponding sets contain a point. So here, there's a space that we can build. It's called a nerve complex, a certain covering of that space. Notice that we've recovered the circle itself, except we've lost this curvature. We've lost a little bit of detail. So the idea is, let's do the same thing for point clouds. So suppose I'm given a point cloud and a covering. I can build a simplicial complex in the same way. But in this case, the one thing that I said in that development was I took connected components. Okay, for data sets, I can't take connected components because, in fact, they break into individual pieces all by themselves when they're finite and discrete like that. So instead, we're going to have to replace taking connected components by a clustering scheme. And the particular one we'll use is single linkage clustering with a particular heuristic on the threshold. But that clustering is the statistician's version of doing connected components. How do you choose the coverings, though? By itself, that sounds like a daunting task because that means selecting subsets of the data set. There's a simple systematic way of doing so. Namely, if I have a reference map to a space which has a good covering, then I can take the preimage of each of those sets in the covering and get a covering that way. So think of the real line covered by overlapping intervals. Okay. So the point is you would want to have your reference space to have useful families of coverings attached to it. And so here's a schematic of that. The complex data set is up top. And what you can see is there's a reference map down to the real line with alternating red and yellow intervals. We'll take their pullback to the space on top. So you see we've now got up here, here's a red set. That's the preimage of this interval. And it's represented by this node. Here's this interval. Its preimage consists of two chunks, and we've got the two yellows and so forth. So down below here, I have this simplified representation of this larger complex data set. Okay. So what does one use for the reference map? We typically use a lot of things that make statistical sense: density, centrality. One can do other things, though. One can get eigenfunctions, Laplacian for the Torus-Rips graphs, principal component or multidimensional scaling, which are machine learning kind of coordinates that get constructed, or even particular fields in your data. Okay? This whole construction, by the way, is very much motivated by notions from Morse theory. The idea of taking the connected components is like building some of the cells in a Morse complex. Okay. So now what I want to show you is a series of examples to illustrate how this kind of modeling can work. So first of all, here was our sanity check for the method. Okay. So let me ask you to pretend that you're colorblind for a moment and just look at the upper left-hand network there, but ignore the color for the moment. Okay. What you see there is a network that's got one of those Y-junction structures to it. And this is a data set coming from a small diabetes data set constructed at Stanford some 40 years ago. Okay, 145 patients, 6 fields, one of them being a normalized notion of weight. But what the structure suggests here, you see, is that you have this T or Y situation. It suggests 3 different populations within the data set. Okay? And so what you can then very quickly do is you can then ask yourself, you can select each of these flares that are coming out and ask yourself, what are the variables that characterize those flares? Notice this is turning the question around. Typically one starts with the variables and asks which ones are correlated with an outcome. Here you're saying, actually, I believe that my data set is going to be somewhat heterogeneous. And so I'm going to say, I actually find these groups, and I will then characterize those by some variables. So now we can go back and think in terms of colors, because what I find is that when I asked for the explanatory variables on this group here, the main one that comes out is blood glucose. Not surprising. And by the way, there are no new findings on diabetes here. As I said, this was a sanity check for us. And so when you now color the network by glucose level, you see that indeed this is the high glucose flare, and the other ones are not so high. Similarly, this flare down here is characterized by insulin response, which is another one of the metabolic variables. And then finally, this group up here is not characterized so much by anything. Nothing stands out as characteristic of it. Now, the data set came with a breakup into populations, so healthy, pre-diabetic, and overt diabetic. Weren't used in building the network, but what you can do is color the network then by presence of people in those different groups. And so you see the healthies are here, pre-diabetics down here. It's hard to see maybe, but the red nodes are down here. And then finally, the overt diabetics are clearly up here. So the point is we've used this modeling to recreate automatically a taxonomy for the patients. the same taxonomy that the MDs had come up with. Okay? So now here is another example from a collaborator of ours, David Schneider, who studies disease progression. So he's interested in understanding what happens as you get sick, and then you progress, getting sicker and sicker, and then ultimately start to recover and then become well again. And so what he has done is he's studied many different situations here. He's studied, flu and a number of other diseases, malaria, and created models based on physiological measurements for them. And so what he finds, of course, is not surprising, is he'll find a loop in every case, because what happens, you become sick, you move through the progression. Now, as you return to wellness, you don't go back through the same process as you did when you were getting sick. You return by a different direction. And so he has these models that have been built based on this methodology for all these different diseases. The thing about it is that the time variable in all these things is extremely variable as people go. So in fact, what he finds by using this kind of analysis is that there are certain numbers of checkpoints within each of these data sets where you come and maybe you stay a little while. But this gives him an invariant way of seeing where is the patient in the process of healing or getting better, and therefore is able to sort of take measurements, gene expression, and other things like that to start to understand how that expression is varying as you move through the healing process. Here's another example. This is with Vijay Pandey's folding at home group at Stanford. So this was a data set for a relatively small size organic molecule called an RNA hairpin. They're doing molecular dynamics simulations on them. And so the data set consists of conformations of the molecules that come in these simulations. Now, so using a version of density as that projection that we talked about, this network is what comes out. Now, you'll notice here that the structure of the network is pretty simple. It looks like a line, except there's this little stuff going on right here. Okay? Now, this construction, this modeling construction I'm talking about, is perfectly capable of producing artifacts. And so if I was not able to go back and talk to the scientists, I would probably not put a lot of time into this particular data set. But if we can go back and ask them, we can say, look, here's something that looks kind of interesting. Do you see anything interesting in the difference between these three? And indeed they did. They found that those corresponded to 3 different trajectories to the folded state, which is one of the main things that they were looking for. The point about this is that this data set actually is quite large, and the area within that data set in which these different trajectories occur is very, very small. So if you try to do principal component analysis or multidimensional scaling on this, you won't find that level of detail, and you also won't see it with clustering methods, because the data set doesn't really break up overall. It's just in this one little region that it does. And so the idea is that you can locate detail within a much larger or more complicated data set. Here's another one. This is a breast cancer data set, gene expression. This is one of the benchmark data sets, in fact, in breast cancer. And what you'll find here is that there's some healthy tissue over here. As you move into disease state, you find that there's a split here. This is so-called basal-like or estrogen receptor negative. This is one of the very bad outcome situation of a well-known piece of the taxonomy. Down here, though, as I get past this area and look at this region here, which is about 8% of the data, it turns out that everybody in there survived the full length of the study. So what you've done is you've located this group in here with a very characteristic kind of gene expression profile, which is very characteristic of survival. Now, so this kind of analysis is interesting. Here is the way that this is often analyzed, is by using dendrograms and hierarchical clustering. So here what you can see is there's a high-level breakdown into 2 pieces. That's correct. That's consistent with the picture that we had. But then now there's a breakup like this with into a bunch of pieces, and we've colored the members of that perfect survival group in red. And so what you see is it's all over the map in that one higher level group. It's not, it's not localized the way it is in the other picture. The reason for that is that clustering has only one thing that it can do, namely it breaks things into pieces. So when you start to apply that, if you have a data set that actually is not breaking into pieces in a natural way, but is really quite continuous, you will very quickly be breaking things apart that should not be broken apart. And that's what's happening here. You're breaking, you're breaking things apart that really belong together. Okay. And the second take on this, this is another more supervised study here. So in this case, we've broken apart the survivors and the non-survivors. We've also added a second data set. So here is this data set that I talked about before, the so-called NKI data set, which we just saw pictures of. And these are the survivors, and then this stuff up here are the non-survivors. This is done using an entirely different technology, where the output, in fact, is completely different. It's different coordinates, different choices of genes and gene sets, and so on. So they have nothing in common. The only thing one uses here is the similarity measure. And what you'll notice is that, in fact, the pictures, though, nevertheless, are very much the same. The shape is here. These are the survivors, or in this case, the no relapse people. And then up here are the clusters of people who have had relapse. So in fact, not only do these two pieces correspond, and these two, but the clusters here actually have a very good correspondence with the clusters up there. So the point is that when you look at just the distance measure, the dissimilarity measure, what that allows you to do is to see the shape of breast cancer itself, as opposed to breast cancer as seen through a particular kind of chip or a particular kind of technology. And that's the value of the coordinate invariance. Okay, now I am running out of time here. Let me say one more, and then we'll move to the next thing. The other thing that we found is that one can look for gross features like flares and loops, and that is kind of our natural thing. That's what we think about. But in fact, having this point of view on it, actually, sometimes you see things you didn't expect. So this thing right here is a picture of a data set coming from a collection of sequences using so-called copy number variation. And it's done from 3 separate populations: Han Chinese, Igbo tribesmen in Nigeria, and Caucasians in Utah. Now, any method that calls itself a method for data analysis can find these 3 groups, can break into these 3 groups. That's not a hard problem. But if you look at them a little bit more carefully, what you'll see here is that the structure is slightly different. Here, this is kind of carpet-like with a relatively uniform degree. Over here, it's much more stellated. You've got certain central attractor nodes, if you like, and then a lot of connections to them occurs in both of these. So you might think this has something to do with the biology, but in fact, it does not. In fact, what it has to do with is the nature of the study. So in fact, there was a processing step taken in the case of the Chinese sequences that removed parent-child pairs, that said, if we have a parent and child in there, we're going to remove one of them so that we have kind of a more uniform representation, not having to do with the family structure. Whereas that step was not taken in the case of the Nigerian or Caucasians. Another method that one can use this, we typically think of building the data set out of the rows of a data matrix, which gives us our distance function. But there's also value in studying the columns of the data set. And so in this case, this was a data set whose rows were time points, and where the columns were various economic indicators. And the question was, can one, for a particular business, study correlation of economic indicators with business performance or with revenue? And so what you can do is you can build a network of the economic indicators, which is what we've got here, and then color it by correlation with revenue for a particular business unit. And so in this case, what turns out, you can see there are sort of 5 hot spots here for correlation like that. So a modeling step, and this was actually critical for the people who were doing this, that sort of just consists of choosing a certain number of indicators, says, let me just choose one indicator for each of these, and I'll get a small, compact model which actually performs quite well. Okay. Okay, and then let me talk about this one as well. This is the data in this case, in the case of this data set, it consists of questionnaires filled out by the personnel in an intensive care unit. And so there are various questions in there about the patient. As each patient comes in, you fill out these questionnaires. Collaborators of ours had built, using genetic algorithms, a model for predicting that. And in fact, the upper picture here now is the network built on that same data and with the predictor. And you can see there's a nice smooth movement from blue to red here. Blue means ready to go home. Red means at severe risk for, for death or other things. Now, what you can also do is color by actual survival, ground truth. And so what you find here is that, indeed, the thing behaves reasonably well. It's doing the right thing over here. But there's also a systematic group up here. It's small, but not to be ignored, which is red, and whereas the predictor is green. Okay? So it turns out that you can rapidly explain that. You can find the variables that explain it and find out that it turns out that in this group, there was a family of questions that were not filled out in the questionnaire, and they concerned the mood and the energy of the patient. So now, of course, having seen that, you can take a step where you would build other, you would modify your model to take into account, are those questions filled out? You'd build a separate model for that to improve your prediction step. Okay, let me now move through the rest of this. Okay. So I talked in the beginning about representing shape and measuring shape. So what do I mean by measuring shape? Shape is, after all, a nebulous concept, but it is something important, as demonstrated by some of this discussion up here. It's important to make it precise. So we'd like to try to measure it precisely in an appropriate sense. So it turns out that the way you do that is you achieve this by trying to count occurrences of patterns, but in an appropriate sense. And let me show you some pictures to illustrate what the difficulties that come up when you do that are. So here's a shape. So imagine that the shape is the black object. The white things are obstacles there in the middle of it. How am I going to measure the presence of obstacles? Well, here's an experiment I can do. Supposing I have a lasso, and I'm just thinking formally here. I throw the lasso out in my field here, and it stays in the two-dimensional field. And now I try to pull it tight. And if the whole lasso comes back, I know there was no obstacle in the middle of it. But if I get stuck, if I get hung up, then indeed there's an obstacle. Okay? So that's sort of a first hint at how one might attempt to measure the presence of an obstacle. Okay. But now here's the thing. That's a very weak thing. It says, is there or is there not an obstacle? Ideally, you'd like to be able to count the obstacles. Okay? So let me point out that the lasso might be thrown in such a way that it loops twice around it, and if I pulled it, I would still get hung up. I couldn't tell the difference between the two. Second point is that I might throw the loop out even in the same way, but just slightly perturbed, and I would arrive at the same answer. So I can't ever actually count all the possible ways of throwing the lasso, all the possible loops. Though that's going to be an infinite thing for any problem of interest. Okay? So what I have to do instead is I have to say, well, I have to understand that some of these loops should be regarded as the same or equivalent. And so here, if I can fill in a surface between the two loops, what I'll find, I'll consider those the same. And so what I might hope to count then is the equivalence classes of such things, the equivalence classes of such loops. So this might be a different equivalence class. And now it turns out that I can actually add the two loops together as well. As you can see here, I can throw one out and then throw out another one, hook them together at the base at a base point. Now here's the thing. Because of this example, because of this example here, you'll notice that actually even the counting of the equivalence classes is an infinite problem, because I can have something that loops around twice, 3 times, 4 times, 5 times. Those would all be distinct. And so actually, even the equivalence classes are infinite to count. The reason one has to introduce algebraic topology then is the fact that it turns out you don't want to actually sort of naively count things. You want to recognize that they are an algebraic object in which you can add things, and in fact, count the dimension of that thing. So in other words, not count the individual occurrences, but count the number of parameters involved in parametrizing the loops. And that's what happens here. So you can actually parameterize these loops by pairs of integers in this case. Okay, so it performs counts of occurrences of equivalence classes of geometric patterns. So what comes out of that process, and by the way, there's a lot of precise mathematics behind it. I talked about lassos and pictures and pulling things in. That doesn't sound very precise. In fact, there's very precise mathematics, basically linear algebra over the integers or over finite fields that allow you to do the following. They allow you to construct, as dimensions of vector spaces, Betti numbers. So these are numbers that count features. So Betti naught is the number of connected components. Betti 1 is a number of loops suitably defined. Betti 2 is a number of two-dimensional surfaces, and so on. You'll notice here that Betti 1 is 0 because, although there are many loops on the surface of the sphere, they are all equivalent to each other under that notion of equivalence that I talked about before. Okay, so turns out huge contribution was made by Emmy Noether, who recognized that this counting problem of trying to count holes and so on was really a question about dimensions of vector spaces. Now, that doesn't sound like a big deal because we know that vector spaces are all parameterized by their up-isomorphism, by their dimensions. But what she observed was, furthermore, that these vector spaces are functorial, that they have the property that when there's a continuous map from x to y, there's also corresponding linear transformations on homology groups. And those things, that fact is sort of key to all the applications and all the computational methods that exist for algebraic topology. So the functoriality, innocent as it sounds, is a huge, huge deal. Okay, now I'm just going to talk about how they're defined linear algebraically like this. Here's a simple description of a space. I've got 3 edges and 3 nodes. I will build a matrix, boundary matrix, which puts a 1 in whenever the node in the left-hand column is in the edge on the upper side. So the properties of this boundary matrix actually allow you to actually do the connected components by taking the quotient space by the columns. And the null space turns out to be one-dimensional and represents that cycle in there. So this little observation here about the correspondence of the properties of this boundary matrix with the geometric properties of the complex can then be embellished and made much bigger to capture all sorts of higher dimensional behavior, and that is what homology is about. So the idea is now to try to extend homology to a much more general setting, that is to say, where we don't have complete information, where we don't have the complete space, but we just have a sampled form of it, a finite metric space or a point cloud. So the method is called persistent homology, and it's what's been developed over the last 10 or 15 years. So how do you do it? How do you define homology? How do you to point clouds sensibly? Because finite sets are discrete. You can't say anything about them just by taking their naive homology. But statisticians kind of knew the answer. They knew what to do long before this business got started. Because they knew this. So they said, look, suppose that I have a finite set equipped with a distance function. That is, input to a lot of statistical questions that one can ask. They said, look, let's do the following. For each threshold r, let's choose a number r, and let's just connect into clusters any 2 points which are a distance less than r apart, and take the equivalence relation that generates. That produces a clustering. But not only that, as then r increases, the clustering becomes coarser and coarser. And so what they did, they said, look, maybe I have some good heuristics for choosing that threshold r, so I know what the right answer is. But actually, I don't really believe that, because I find that I build some heuristics, and as soon as I do that, I try it in some other new situation, and it's not right. I need to fiddle with it. How about instead if I have a way of understanding the behavior at all thresholds all at once in a single profile? And that's what this is. That's what the dendrogram is. Because you'll notice here what the dendrogram says. At 0.6, I can slide over and say there's 4 clusters at 0.6. 0.8, there's 2 clusters, and so on. Not only that, though, not only can I count the number of clusters at each level, but I can also count what clusters at level 0.6 correspond to what clusters in level 0.8, right? So these 2 clusters correspond to this one, these 2 to this one. Just having the numerical thing, it might very well be that this one had connected to this one, and so on. So the statisticians knew this. The big leap that they took was to say that I can encode the whole behavior for all the parameter values in one profile. Okay, so that's the story on that. So now one can extend this. So suppose that one wants to understand more than clustering, more than breaking into connected components. I'm going to actually build some spaces out of this. So here's how I can build the space. I have the I can take a radius r, and I can take the balls around that point and understand their overlaps, the overlaps of those balls. But maybe a better form of it is this. Let's notice here that I can build a space in that way, as I just described, or build a complex. But what's important is that when I build that for varying thresholds, I actually get maps between the spaces. So this is a small threshold, and the complex that's built is 4 distinct points. Here's a somewhat larger threshold where I get a loop, and then an even larger threshold where everything is connected and I get a tetrahedron. Okay? The main point is that not only do I have these spaces, but I have the maps in between them. And so actually I can apply Emmy Noether's functoriality idea to homology now, which counts loops instead of connected components, and start to make sense of that. So here we go. We get a diagram of vector spaces, and such structures are called persistence vector spaces. Okay, that's the name that was given to them. Now, ordinary vector spaces are classified by their dimension. Very easy to understand. Question is there a corresponding construction or a corresponding parameterization of the isomorphism classes of these persistence vector spaces? And the answer turns out to be yes. So it turns out that the classification is parameterized by so-called barcodes, which are finite collections of intervals, unordered. The order of them makes no difference. And it turns out that these are readily computable due to sort of judicious use of abstract algebra. The main point, the main takeaway is that what you get out of this is when I have a data set that looks like this, which I think we all would agree looks like a circle, I get the one-dimensional barcode that comes out looks like this. So it's a finite collection of intervals, but what you'll notice here is there's a bunch of little ones and then one long one. So the long one is the signal for the presence of the big loop. This stuff in here, this is stuff that happens as this complex arranges itself. You'll see some small loops which are born and then die again very shortly. But the overall signal here is this one right here. Okay? And just to give you another take on it here, why it's called persistent homology, here's a data set. I think we would agree that this looks like a circle. In fact, though, because when I do the homology, homology doesn't see the difference between large elements, large loops and small loops. So here, what we'll see is Betti 1 equals 3. The big loop, which is there, that's a good thing. These two smaller ones are there. That's not so good. We really believe those come from noise or something along those lines. But you might say, well, look, you just had the wrong heuristic for choosing the threshold. So why don't you just enlarge the threshold and you'll fill in those holes and everything will be fine. So I can do that, but then what happens is I get the thing at the bottom here where there's an edge, a bridge that crosses, and so here I get Betti 1 equals 2. So in other words, I don't win either way with either choice of threshold, but if I take the point of view that the geometric stuff corresponds to those things which persist across the change of scale, then I'm in good shape. And that's the idea of persistent homology, because the big loop is the only thing that transforms, that comes from the one below and goes in a non-zero fashion to the one above. Okay. So I just want to very quickly, because I know I'm about at the end of time here, talk about a couple of examples of this. Oftentimes in phylogenetics, one is studying data sets coming out of that, and the assumption is that they should look like trees. But in fact, they're not always tree-like, and so the persistent homology, it turns out, gives a way of assessing how tree-like space is, because in fact there's a theorem to the effect that the persistent homology in dimensions greater than 0 actually vanishes for trees or even subsets of trees. So in work with collaborators at Columbia, we actually studied a number of these viral evolution data sets and found that in many cases the tree-like hypothesis did make sense. In fact, that it did come out with vanishing homology, but also in other situations it did not. And it turned out then that in fact, what was very surprising was that the presence of a loop like this, okay, you could actually pull out the generators for that. So you can pull out the parts of the complex that were involved. And those turned out to then give information about alternate mechanisms for evolution that happen. So it turns out when we think of evolution, we think of it as, it's done by reproduction. It's what happens maybe through mutations. But in fact, for viruses and other organisms, there are other mechanisms that sort of take more genetic material at once and perform more complex changes. And so what the homology in this case turned out to reflect in many cases was exactly the presence of those alternate methods of evolution or of genetic change. Okay, now here's another one, and I'm going to have to run through this very quickly. This was a data set now that came out of the following. One of the things that one can do is if you're studying databases of images, you can ask a much more theoretical question about those as to what characterizes images within the set of all possible assignments of grayscale values to pixels. So we're thinking here of digital cameras with grayscale values at each pixel. So this space is very big, the space of all possible assignments of grayscale values. But David Mumford asked the question, what can you say about the set of all images inside the set of all possible assignments of pixel values? At the end of the day, he came to the conclusion that, certainly the normal mode that a topologist or a geometer would think of this is not really appropriate here. That is to say, the space of images is not like a submanifold. It's not probably something that's very easily studied, just because it's of extreme high dimensionality, many other things. But what he said was, look, let's do something simpler. Let's study the local structure of the images statistically. So specifically 3x3 patches like this. So what they observed is that each patch gives a vector in R9. Most patches will be nearly constant because, in fact, there are typically large solid regions in images. So the low contrast things, or nearly constant ones, will dominate the statistics. We'll remove those and study only the high contrast patches. Contrast they measured in a certain way. They built off a data set constructed in Holland, a data set of a few thousand images. So from that, they constructed then about 4.5 million high contrast patches from a collection of images by these two Dutch neuroscientists. Let me sort of move through this rather quickly and say that we did the following. Our take on that data set was to do the following. Was to say, look, let's try to understand that data set of 3x3 high contrast patches. And so the naive thing would be, well, maybe we just compute persistent homology of that. They had done some transformations to where the data set lies in a 7-sphere, out of those 9. So there was a couple of dimensionality reductions that they had done. Unfortunately, you find out that everywhere in that 7-sphere, there's a data point nearby. Okay. So, in fact, the best you could hope to do if you did that was to actually recover the homology of the 7-sphere. And computationally, that would be a difficult thing. But even if you did, that wouldn't be very interesting. So it turns out, though, that even though there are there's a point everywhere in the 7-sphere, the density actually varies a great deal. So, in fact, what we're going to study is the high-density part of the high contrast part. And so this is what comes out. So here's this was a downsampling of 50,000 points. You'll notice here that there's a bunch of short intervals. That happens as the construction is arranging itself, but then there's a long line here. Now, that's not so hard to understand. Question is, what does this suggest? It suggests that the data set is concentrated around a circle, but in fact, it's not so hard to understand why that is. It's because of this picture. As you can see, it's moving around. It's a single linear gradient going from dark to white. On the other hand, what happens, so let me very quickly, the density estimator that we use, there are various options for that density estimator. Some are more local, or use more local information, and some use more global information. Here's a more local one than that previous one. And in this case, you can see that what you get is something quite different. And you get it no matter, you take many samples and it always comes out the same. It's not just that it comes out to be a lot of bars, it always comes out with 5. Okay? So the question is, what's the explanation for that? And it turns out that a good explanation is this 3-circle model like this. And here you can see that it has 5 loops. So that was surprising to us and suggested that one look for a 2-dimensional version as well. And this is what comes out there. The resulting object is, turns out the geometry of it is a so-called Klein Bottle, which is an unorientable surface. And we have used that to go ahead and do at least 2 different things. One is to build algorithm for texture recognition and discrimination, and another is a compression algorithm for single images. All right, so I'm sorry I'm over time, so thank you very much.